everyone. Continuing with our series in biotechnology, I, Dr. Charu Dogra Rawat from Ramjas College, University of Delhi, would be discussing today about the gene editing tool CRISPR-Cas9. We have already been discussing gene editing tools and we had discussed zinc finger nucleases and tail nucleases in our previous sessions. And today we talk about again a very powerful gene editing tool, CRISPR-Cas9. The learning objectives for today's session would be to understand about the structure, mechanism of action of the CRISPR-Cas9 gene editing tool, to understand the origin of these CRISPR-Cas9 arrays, and to understand the advantages and limitations of CRISPR-Cas9. So let's review a little about what these gene editing means and what are these gene editing tools. The gene editing or genome editing is a method for making specific changes to the DNA of a cell or organism. Now we are doing the series for biotechnology. As I always say, biotechnology is an amalgamation of biology to create some kind of a technology. A major technology is the genetic manipulation, that when we edit or design the genes or genomes according to our needs, so that we can use it in therapeutic purposes or industrial purposes or any other different kind of applications of biotechnology, that is. So this gene editing or genome editing is requiring specific changes to the DNA is a very important technology. It can be used to add, remove or alter the DNA in the genome and based on the engineered or bacterial nucleases. So we saw that gene editing requires either the naturally occurring nucleases or the engineered nucleases with efficient cleavage and cutting techniques. An important aspect of gene editing is homologous recombination. It is the earliest method used to edit genomes in living cells. It involves the exchange or recombination of genetic information between two similar homologous strands of DNA. Now, homologous recombination is a natural phenomenon. You know that it is occurring in the cells during division. For example, in meiosis, when there is crossing over, then the homologous recombination takes place. Or otherwise, for example, there are transposases which comes or get into the different kind of cells and again cause various kinds of homologous recombinations to occur. Now this involves the exchange of genetic information. So there is some kind of an alteration in the genetic content is taking place. So this was basically the initial method to edit the genome, to change the genome in the living cells. Isolated undamaged DNA fragments bearing genome sequences similar to the portion of the targeted genome are injected into the individual cells or taken up by the cells using specific chemicals. So homologous recombination involves exogenous DNA to be introduced into the cell, where it goes, it finds its homologous sequences in the targeted genome, and then the homologous recombination occurs and the exchange of genetic material takes place, creating or synthesizing or causing the insertions, deletions, recombination, etc. Once inside a cell, these DNA fragments can then recombine with the cell's DNA to realize the targeted gene addition, replacement or inactivation. So this was the initial thing, that the homologous recombination, which is a natural phenomena, was employed to create the genetic editing by introducing the exogenous DNA. This is how it is depicted in this picture. Then when this is, for example, the target genome, and this is for example the vector which is there. So this is the genomic DNA, this is the targeted vector. Now it is having some homology. So these are the homologous sequences to the genomic DNA. When this exogenous DNA containing the gene of interest or whatever manipulation we want to do, not just inserting but even deletions or additions or the recombinations, whatever is there, there needs to be a homology with the genomic DNA, and then by homologous recombination it will be, for example, inserted, or there can be excision that can take place by removal. So there can be additions, there can be deletions, there are various outcomes of this kind of a homologous recombination. So there can be insertions, integrations, replacements or deletions that can occur. So this was the basic mechanism of homologous recombination. Homologous recombination gene editing, however, had to be improved and newer techniques needed to be found because it is extremely inefficient in most cell types. First of all, introduction of exogenous DNA to all the cell types is not really very feasible, for example, and then HR gene editing is not very targeted. There needs to be some kind of a homology as well. So it is an inefficient in most cell types. It is inaccurate and has a high rate of error when the injected DNA fragments insert into an unintended part of the genome, causing off-target edits. So off-target edits is again a bigger problem, because this homologous recombination gene editing is relying on the fact that whatever we want to add or wherever portion we want to excise, then there needs to be some kind of a homology. Now that some kind of a homology, there can be multiple places where the homology is there, except for where we, where our desired target sequence is there. So there is a lot of inaccuracy that can occur and a high rate of error can occur. So another thing was that the generation of nucleus induced double stranded breaks leads to the stimulation of highly efficient recombination mechanisms of cellular DNA. So it was observed that the homologous recombination involves the creation of the double stranded breaks. Now these double stranded breaks therefore can be introduced by many different ways. So we can have certain nucleases which cause these specific double stranded breaks, and therefore it can stimulate the efficient recombination mechanisms. Nucleus induced DNA double stranded breaks can be repaired by one of the two major mechanisms that occur in almost all cell types and organisms, which is the homology directed repair and the non-homologous end joining method, resulting in targeted integration on gene disruptions respectively. Both these mechanisms have been talked about in detail in our previous sessions of gene editing tools. So I am just reviewing these things, so I am not going into the details at this point of time. But these double stranded breaks that are either created naturally in the natural homologous recombination mechanism procedure, or if we introduce a certain nucleases that can cause these double stranded breaks, then after these breaks, these two repair mechanisms repair it, leading to these genome editing outcomes. So there can be actually the gene knockout, where the gene can actually be inactivated or just removed. There could be gene inhibition, there could be gene inversion. When the gene is there, then you can cause the homologous recombination, and when the repair occurs, the gene orientation is actually reversed. That can be there. Then there can be also be certain kind of gene additions, or there can be gene deletions or gene mutations or gene reversions, mutation reversions. All these are the outcomes of the genome editing that we intend for to create in a particular cell of interest, for example in therapeutic cases, or as I said, in industrial production cases, etc. We talked about the various gene editing tools. The initial ones were the large homing endonucleases. These nucleases were used to create the double stranded breaks. So the idea is to create a double stranded break, and these double stranded breaks will then be repaired by the indigenous repair mechanisms. Now creating these double stranded breaks, again comes the specificity, so that there are not many off target edits which are there. So these homing endonucleases were there. Then came the zinc finger nucleases. These has been talked about in detail in our previous sessions. You can please refer to that. So there are zinc finger nucleases which are there. Again, they contain FOX1 endonucleases and then zinc finger like proteins, DNA binding domains. So DNA binding domain gives some kind of a specificity where it is binding, and then FOX1 endonucleases are creating the double stranded breaks. Then we also talk about tail nucleases, where they are again the FOX1 endonucleases was used, and they have these tail proteins associated with it. So these tail proteins again provide the specificity that where these molecules are going to bind, and then the FOX1 endonucleases are going to cause the cleavage or create the double stranded breaks, which can then be repaired and leading to the genome editing outcomes that we intend to attain. Another system that we discuss in detail today is the CRISPR-Cas9 system. Now what is this CRISPR-Cas9? CRISPR stands for clustered regularly interspaced short palindromic repeat, and that is what CRISPR is. So, CRISPR, when we say clustered, so it is not a single unit, it contains a cluster of unit, regularly interspaced. So, these clusters are regularly interspaced by certain spaces, and they contain short palindromic repeats. So, it is a cluster of short palindromic repeats which are regularly interspaced by certain spaces. That is what the CRISPR is, and we will look at its structure and mechanism of action in detail. This CRISPR gets associated with a protein which is called as Cas9. Now, there are a series of Cas proteins which are there, and they have been divided into 6 categories. The CRISPR-Cas9 system belongs to the type 2 category, and we will discuss only CRISPR-Cas9 today. So, there are many other orthologs of Cas9, or there are many other Cas proteins that have been also discovered and used for various purposes, but today our focus of discussion would be only the CRISPR-Cas9 system. So, I hope you understand. So, really you do not have to mug up or remember this thing, but you can also derive this thing if you have the understanding of what CRISPR is. So, CRISPR is the clustered regularly interspaced palindromic, short palindromic repeats sequences which are basically there. Now, this CRISPR-Cas9, let's look at some of the characteristics and then we'll talk about it in detail. The recognition site is a single stranded guide RNA. When we talk of these nucleases, or when we talk of these systems, when we want to do the genome editing, there are 2 aspects to it. One is the recognition of a particular site, and the second is the creation of the double stranded breaks. This is true for all the tools that we have discussed in gene editing tool. So, this specificity, that where our target is, how it is going to go and bind to that target, is that one aspect that needs to be present as a characteristic in the CRISPR-Cas9. So, in this case, in case of genome editing tools. So, in case of CRISPR-Cas9, the recognition site is a single strand guide RNA. We will discuss about this. The modification pattern is basically a cell nuclease. So, that is what it contains. The target sequence size is typically 20 base pairs guide sequence and plus a PAM sequence. So, this guide sequence, this guide RNA sequence is approximately 20 base pair in size, and then it also the recognition, the target site should contain a PAM sequence, which is basically we will discuss about again what this PAM sequence means. But this is the requirement for the CRISPR-Cas9 system. The specificity is that generally it is very specific, but it is usually tend to be moderately specific and does create some kind of an off site targets as well, but there are different orthologs that have been discovered to Cas9 which increases the specificity of this particular system. So, what is the targetic limitations? As I said, that target do not proceed without a PAM sequence. It requires this PAM. So, this is a limitation, that you cannot edit any sequence, but you need to have this PAM sequence above the sequence that you want to edit. So, this is a particular limitation that is there. As far as the difficulty of engineering is concerned, it is not a very difficult process. It uses the standard cloning procedures and the oligosynthesis. So, this is a routine thing that happens in lab. So, the difficulty of engineering is not that high. As far as the difficulties of delivering, because when we are creating this kind of a system, we need to deliver it into the cells that needs to be edited. The difficulty of delivering for CRISPR-Cas9 is moderate, as for example, the Cas9 is quite large in size, but as I said, there could be different orthologs that can be used, which increases the efficiency as well as the specificity of this CRISPR-Cas9 system. So, that is the basic characteristics of these CRISPR-Cas9. So, this is just an overview to show. We will discuss about this in the mechanism of action, but the basic thing is that this is, for example, the genomic DNA, and this is the part that you need to edit. You want to edit this part. So, you will have a CRISPR-Cas9 molecule, this kind of an arrangement where you will have a Cas9 protein, and this Cas9 protein will be associated with a guide RNA. So, this red region is a guide RNA. So you will have this guide RNA, and in this guide RNA there will be a portion that will be complementary to the target site what you want to edit. Now an important aspect, as you see here, this black is the DNA that you need to target, and this is what is called as the sgRNA, or the single stranded guide RNA. So as I said, this guide RNA will have a sequence that is specific to the target. So this CRISPR Cas9 molecule, this Cas9 molecule will be guided by this guide RNA to go and bind to the target by complementary binding, and then this is basically going to cleave it at, for example, this red portion which is adjacent to the PAM sequence. So PAM sequence is very important for the target site to be there. The Cas9 nuclease from Streptococcus pyogenes, now it has been isolated from other organisms as well, but initially it was from Streptococcus pyogenes, is targeted to genomic DNA, shown for example in the human EWX1 locus by a short guide RNA, sgRNA, consisting of a 20 nucleotide guide sequence and a scaffold. So this is the scaffold, this is the guide sequence, this is the sgRNA, and this Cas9 nuclease is targeted to the genomic DNA, and it is targeted to this particular genomic DNA as shown in the figure. The guide sequence pairs with the DNA target directly upstream of a requisite 5' NGG adjacent motif, which is termed as PAM motif. Cas9 mediates a double-stranded break approximately 3 base pairs upstream of the PAM region. So this is what a molecule basically looks like. We'll talk about its mechanism, but this is how it looks like. So this is the target DNA, this is the short single-stranded guide RNA. This guide RNA directs or guides this Cas9 to bind to this particular target. The important aspect is there needs to be a PAM sequence, and the target site needs to have the complementarity with the guide RNA. So it will create a break, and now the double stranded, this Cas9 is a nuclease, it is going to cut 3 base pair upstream. So this red part where it needs to make a cut and create a double stranded break, which can then be regularly, routinely repaired, etc. That can happen. So this is what the CRISPR Cas9 is basically looks like. But let's have a look at the CRISPR locus first. So this CRISPR locus, in genomic DNA, you have got Cas9 genes operon, which is present at one end, and then this is the CRISPR loci, how it looks like. Now you can see there are different colored blocks that are shown here. So these different colored blocks are basically that there are certain repeat regions which are present. So there are repeat regions which are occurring regularly after a certain distance in the genome, and in between interspersed is the spacer sequences. So this is how the CRISPR loci look like. It has Cas genes. Now these Cas operons can produce a wide variety of Cas proteins. As I said, there are 6 categories of Cas proteins, again containing many different variants. What we talk about is the type 2, which is the Cas9 system. Again, the mechanism of other Cas9 proteins differ to some extent, but that is how basically it looks like. So we'll discuss about the Cas9. And this CRISPR RNA, this array which is there, is transcribed to give rise to this pre-CR RNA, which is shown here. So how does the CRISPR locus or the structure of the CRISPR loci looks like? Each system comprises a cluster of CRISPR associated genes. So Cas stands for CRISPR associated genes, some non-coding RNAs, and a distinctive array of repetitive elements termed as the direct repeats. These repeats are interspaced by short variable sequences. So this is what we have been discussing. This is picture from another source, but again it is basically showing that this is the CRISPR these are the CRISPR associated protein genes. Then there is some leader sequence, and then for this is a CRRNA will be transcribed from this particular genome site. As you can see, there are direct repeats which are present, and in between these blue colored direct repeats there are different spacer DNAs which are located. Now where does these spacer DNA comes from? These sequences are derived from exogenous DNA targets known as protospacers. For example, if a bacteriophage infects a bacteria, a prokaryote, then it injects its DNA, then a piece of its DNA termed as the protospacer is introduced into this CRISPR array, and that is basically creating the CR array containing the direct repeats, and in between the exogenous DNA, for example, that has come from a bacteriophage, is introduced in these spaces. So these are variable sequences, because one time one bacteriophage infected a particular bacteria, it created this short fragment and introduced it into this particular CR array, which is said to be. So it is just introduced, so it just keeps it there. So these sequences are derived from exogenous DNA targets known as the protospacers, and together they constitute the CRISPR RNA array or CR-RNA array. So I hope you understand the structure of the loci. I just review once again that there are certain Cas genes which are located. Now it can be located at a distance from this particular CRISPR array. The CRISPR or CR-RNA array basically contains a direct repeat, series of direct repeats interspersed by these spacer elements. Now these spacer elements arise from the protospacers that has come from the exogenous DNA. So I hope you understand the structure of the CRISPR loci. Now what is the use of this CRISPR loci? We are harnessing it for biotechnology, for developing a technology or genome editing tool. But why was this a natural phenomena that was occurring in bacteria? What is the significance of this CRISPR-Cas system to the microbial cells from where they have been isolated? This CRISPR-Cas is a microbial adaptive immune system that uses RNA-guided nucleases to cleave the foreign genetic elements. So this CRISPR-Cas constituted the immune system of the microbes, because they can use these RNA guided nucleases to cleave any foreign genetic element. How does it do that? Upon infection, the new foreign DNA sequences are captured and integrated into the host CRISPR loci as new spacers, like we just said. So what these spacers are, they basically act as the memory of the infection that has occurred. So this is how basically it looks like. So when a bacteriophage, for example, infects a bacterial cell, or any exogenous DNA is taken up by the cell, then these protospacer elements are introduced in between these direct repeats and they stay there. So they are like the memory of this particular infection. When they will be transcribed into single guide RNA, and this RNA will then combine with the nuclease molecule to create this CRISPR-Cas system. It will occur when there is a second infection, for example, of the same bacteriophage. Now this particular CRISPR-Cas9 molecule has a spacer element which is homologous to the DNA of this bacteriophage, because it has infected earlier, it has kept a short piece of it. Now this short piece is homologous to its DNA. So this CRISPR-Cas9 is going to target the second infection introduction of the DNA, and it is basically going to cleave it. So this is how the adaptive mechanism it uses to defend itself. Spacers serve as a genetic memory of previous infections. If another infection by the same virus occurs, the CRISPR defense system will cut up any viral DNA sequencing matching the spacer sequence. If a previously unseen virus attacks, a new spacer is made and added to the chain of spacers and repeats. Within the DNA target, each protospacer is always associated with the protospacer adjacent motif or PAM, which can vary depending upon the specific CRISPR system. So for example, for CRISPR-2 system, we said it is 5' NGG. So that is the PAM motif for the CRISPR-Cas9 system, type 2 system. So this within the DNA target, this PAM is again very important. So that is how basically the structure of the CRISPR-Cas9 loci is. We discuss the mechanism of its action and its advantages and limitations in our next session. Thank you.